Alright, so this is 12, this is biotechnology. The biotechnology is a broad field that is used to analyze a variety of macromolecules in various different ways. But the molecule you guys chose to focus on in this particular lab was DNA. So we focused on DNA in this particular lab. Now, I don't want to go too much into the structure of the molecule of DNA, but I do want to kind of give you enough of a background on this molecule so these experiments at least make sense. First and foremost, DNA is what kind of biological macromolecule? What class? Nucleic acids. You may recall that nucleic acids are made, their monomers, which are called nucleotides, are made from 3 subcomponents. One of them is a sugar. One of them is a nitrogenous base, the A's, C's, T's, and G's. And one of them is a phosphate group. And what I ultimately want to remind you of is that if you actually look at a molecule of DNA, what you would see is the following. This needs to be a part of the technical here. All right. Go. Okay. Now, I don't want to spend too much time on the structure, but the one thing I want to point out to you all is you'll notice that in DNA we have what's known as a sugar phosphate backbone, and if you focus in on those phosphates, what you'll notice is that each phosphate has at minimum an oxygen that has a slight negative charge, or in this case, a negative charge. So if I've got a bunch of those phosphate groups on a molecule of DNA, overall, do you think that that molecule of DNA is going to be positively charged, negatively charged, or neutrally charged? Probably negatively charged, because we've got a bunch of negative charges. Keep that in mind, because it's going to come back at one of these experiments. Okay? So, the first thing we want to do, or that we wanted to do in this particular exercise, was we wanted to isolate DNA. So our first experiment was DNA isolation. What organism did we use for this? B-A-N-A-N-A-S, we used bananas. Yes, okay. We used bananas. So you crushed up your banana, or blended it, with 250 milliliters of water. Now, let's take a closer look at a banana cell. So a banana cell, which is a plant cell, has a cell wall and a plasma membrane, and where is the DNA going to be? Inside the nucleus, right? It's all the way inside that nucleus. Now I want to isolate the DNA, which means I need to get it away from everything else in that cell. The cell wall, plasma membrane, nuclear envelope, and then also in the nucleus, Gesundheit, in the nucleus, what is that DNA also wrapped around? Histone proteins. So, the two things I need to do, I need to disrupt lipid bilayers, and I need to get rid of proteins off of that DNA. So I add 2 different solutions to this. I add 25 milliliters of each, and one of these solutions is going to help me remove the lipids from around the DNA or on the nucleus or the phospholipid bilayer. And the other one is going to help me remove the proteins. What was one of the 2 solutions we used?
Meat tenderizer.
Soap. I used soap and I used meat tenderizer. I believe the percentages, if you're into that kind of thing, were 50% soap solution and a 5% meat tenderizer solution. Let's talk about what these do. You've used soap once before. It's an emulsifier. What experiment did you use it in? Macromolecules. You used it with that layer of vegetable oil, and it broke up and separated many of the lipids in that oil. So, soap is going to be used to disrupt the lipid bilayers that we have in this cell. So the soap is going to take away these, and the cell wall is going to be dealt with as well, but it might not necessarily be soap, and I'll talk about that in a second. But that still leaves proteins bound to our DNA. Enter meat tenderizer, which contains distinct molecules called proteases. Somebody tell me what you think these are and what they do. They're enzymes that target proteins. Excellent. By the way, just fun fact, if you ever get stung by a scorpion, throw meat tenderizer on the wound. Trust me, it works. Okay? So, just FYI, there are scorpions here, so in case you ever encounter one, there's what you do. Now, after we added all these together, we also boiled this solution. So we boiled this mixture afterwards, and the boiling is also what helps us break down that cell wall. Now there's one final problem here, and that is that DNA is actually soluble in water, and we're trying to isolate it. So the final thing we did here, in this exercise, the very last thing that we did was we took that mixture and we dumped it into a test tube, and then we had to add a solution that DNA is insoluble in. And that solution is ice-cold ethanol. Once you add that ice-cold ethanol, you should see this white kind of stringy stuff that has bubbles in it. And that represents our DNA. That is our DNA. This is a very rudimentary, but it also is still used in a similar fashion to this. This is the technique that is ultimately used to isolate DNA. Okay. So, we've isolated our DNA, now what? Well, I'm glad you all asked. So after we isolate our DNA, the next thing that we have to do is we actually have to start trying to establish differences between different people. And to do this, we're going to use a class of enzymes that are called restriction enzymes or restriction endonucleases. So let me explain to you what these are. These are enzymes that recognize certain bases, sequences of bases in your DNA. Okay, so maybe AAA TTT with its complementary strand of TTT-AAA. Okay? So they recognize distinct regions. Because we're all unique individuals, the number of these regions that these enzymes can recognize in your DNA may actually differ from mine. So let's say that after the review session, I'm feeling super tense and I decide to go out and, commit a crime. Not that I'm going to, but if, hypothetically, I did, there's a chance I would leave DNA behind at that crime scene. And what forensic scientists would do is they would take the DNA from that crime scene and they would expose it to these enzymes. And these enzymes would recognize distinct regions on the DNA and they would cut it into fragments. So on my DNA, let's say I have 2 of these restriction sites. That will result in my DNA being broken up into 3 different sized fragments. Now let's say that you unfortunately were the schmuck who was in the wrong place at the wrong time. The cops saw you there and they bring you in for questioning and they ask you to submit a cheek swab. So you do, super nervous, but what you can't see is that ultimately it winds up that you only have 1 of these restriction sites. So how many fragments are they going to get from your DNA? 2. Your DNA fragments are not going to match the DNA that I left behind. And as a result, you're going to be released from the police station. So this is how restriction enzymes work. Questions about that? Okay, so now that we've cut our DNA, we need to actually analyze it. And to do that, we're going to perform what's known as gel electrophoresis. So gel electrophoresis is a technique that we can use to separate these different fragments. So, in gel electrophoresis, you have an agarose gel that has these wells. Okay? So it has these wells, and you put your DNA in there. And we want to make the DNA run down the gel. Now, if we just put the DNA in these wells and we let it sit, it's going to start diffusing in all different directions. We don't want that. We want it to go in this direction. So we can exploit a very fundamental chemical characteristic of the DNA molecule to get it to do this. It's going to diffuse, and I'll talk about how these fragments are going to diffuse in a second. But if I want to pull those DNA molecules to the bottom of the gel, I'm going to put a current through this gel. And that's why we call this overall process gel electrophoresis. Okay? Phoresis literally means the process of moving. Electro means electricity, and gel means gel. Okay? So, what kind of charge do I need to put at the bottom of my gel here if I want my DNA to move from the top to the bottom? Why, those of you who said a positive charge, why did you say positive? Because DNA has a negative charge, and what does Paula Abdul say? Opposites attract, yes. So, opposites attract, which means the negatively charged DNA is going to be attracted to the positively charged electrode. Now, if I want to speed this whole process up, what kind of charge do I need to put at the top of the gel here? A negative charge. Good. So, my DNA is going to move through, and going back to diffusion, which fragments are going to have the easiest time moving through the gel? The larger ones or the smaller ones? The smaller. Excellent. So, this band here represents a collection of really, really small fragments of DNA. This one here represents a collection of medium-sized, and this one here represents a collection of larger-sized fragments. So your DNA might actually do something like this, based off of those two fragment sizes. Maybe it might actually even run a little higher here, because it's technically those two things together. But ultimately, what this means is you did not commit that crime, because your DNA does not match it. Questions about gel electrophoresis? Okay, so let's talk now about the two types of analysis you did for gel electrophoresis. Let's talk crime scene, and let's talk paternity testing. All right, so with crime scene, this is CSI stuff. Then once we finish this, we'll jump into blood typing, because I believe that was the last part that y'all had, correct? Okay. So, with crime scenes, you're always looking for a 100% match between the crime scene DNA and the suspect DNA. So I'm going to draw up here two suspects with one crime scene. So we took DNA from the crime scene, and let's say that we digested this DNA with two different enzymes. And typically, forensic scientists do this, because you don't want it to be the luck of the draw that the one enzyme you picked accidentally gives you, two of the same types of banding patterns when the person actually didn't commit the crime. So scientists usually use a bunch of different enzymes. Okay? So we cut up the crime scene here with enzyme A and enzyme B. And let's say the crime scene DNA gives us this pattern. So we'll go off of what I did before. We'll get three bands there. Let's say with B instead, we get something like this. Okay? All right, so now let's do suspect 1's DNA. Digest it with enzyme A and enzyme B. And let's say for enzyme A, I get this. Now right now, it looks like suspect 1 might be the guilty party. But let's say that for the second enzyme, enzyme B, his DNA looks like this. Did suspect 1 do it? No. Even though his or her DNA looked the same when digested with enzyme A, because we get this big difference with enzyme B, that person is not the guilty party. Now let's say we do suspect 2, A and B, and suspect 2 looks like this. Guilty or innocent? guilty, because there's a 100% match when I digest that DNA with enzyme A and with enzyme B. Questions, concerns about crime scene investigation. Essentially what you should do on the exam is start band by band and say, does suspect A have this band? Yes or no? Does suspect 1 have this band? Yes or no? Does suspect 1 have this band? Yes or no? If at any point in time you get the answer no, that person is not the guilty party. Question?
Would a twin have a similar, but maybe not exact DNA?
Twins, if they're identical twins, they would actually probably have pretty darn close DNA. There are still some things that are known as SNPs that occur in identical twins that do make them distinguishable. Okay? So that's crime scene. Now let's go on the Maury show and let's do paternity testing. All right? All right. So we're going to have a maternal lane, a child lane, and we're going to do it very simply, just a potential father lane. We won't do. I can extend it if you need me to. We can do multiple potential fathers. So remember, the way that this works is the child is going to get 50% of his or her DNA from his mother, our mother, and 50% from the father. So that means that if the child only has 2 bands, that child shares one of those bands with his or her mother. And that means that for this person to be the father, they have to share that other band with the child. So if this person instead has something that looks like this, he is not the father, because he doesn't share this band with the child. Now it gets a little more complicated if the child has more than 2 bands. So let's say that the child has 4 bands. The mother and father are both going to have 4 bands as well. And let's say the mother shares those bands with the child. Now for the person to be the child's father, they have to share the 2 bands that the mother does not share with the child. So we would be looking specifically at this band and this band. So again, take the number of bands that the child technically has that are unique to the child, divide that in 2. So if it's 2 bands, the child shares 1 band with each parent. If it's 4 bands, the child shares 2 bands with each parent. If it's 6 bands, how many does the child share with each parent? 3. Excellent. So let's say that we did have several different individuals, and we wanted to know if one of these guys was indeed the father of the child. Let's say that this individual here looks like this. The second individual looks like this. And the third individual looks something like this. Who is the father of the child? 2. The reason it's 2. Okay. Ask yourself, does father 1 have this band? No. Father 1 is not the child. Father 2 and 3 do. But when we come over here, only this gentleman has that band. Now you'll notice that I specifically and purposefully drew in these 2 bands for that third person. The reason why this person is not the father of that child is because the mother already shares those 2 bands with the child. We're looking for the 2 bands that the mother does not share with the child to determine maternity. And so we are able to. So in this case, it would be this individual. Questions, concerns? Shiv?
Like, how do you know that the bands are shared? Like, why can't the bands share? They can't be shared.
How else could this mother give birth to this child? Because this band doesn't match with anything in the child. No, but seriously, if you know for a fact what the actual maternity of the mother is and what chromosome she donated to that child. So notice, the mother doesn't share this band with that child, and the mother does not share this band with that child. If you already know this woman is that child's mother, you start with her first because you know they have to share DNA. We don't know if any of these people are the father of that child. That's why we're not going off of the fathers first and then the mothers. Okay? All right, last thing. In the final minutes, I'm going to do blood typing for you guys. So, let's talk blood typing. the general idea. You have these red blood cells. And these red blood cells can have distinct carbohydrates on their surface. They can have an A-type carbohydrate. They can have a B-type carbohydrate. They can also have an RH-type carbohydrate. Or they don't have to have any of them. Now, in people who have a certain blood type, let's say you are blood type A, what that essentially means is as far as your cells go, you have carbohydrate A on the surface of your red blood cells. There's another thing that it implies, though, for the antibodies that your body makes. If you're blood type A, your body makes anti-B antibodies, which means if somebody who has blood type B gives you blood, those antibodies target and attack the carbohydrate B on that blood type. What we're doing here, though, is we're using this fact to help us figure out what kind of blood a person has. So you had these dishes that had A, B, and RH on them. And in these dishes, you placed anti-A antibody, anti-B antibody, and anti-RH antibody. So those antibodies went to each of those wells. And then what you did was you took the person's blood and you placed it inside of each of these wells. If the person's blood reacts with the antibodies, you get agglutination, which was those particles you saw that started floating in the blood. That tells us that person has that particular antibody. So for example, if I put in A-type blood, or if I put in antibodies against A, and I put in a blood here that I don't know what it is, and I don't see a reaction, does that person have carbohydrate A on their cell surface? No, because if they did, I would expect to see a reaction. So that person does not have A. But let's say here, I do see a reaction, where I see this agglutination. Okay? When I put their blood into this B well that has antibodies against B, what is that telling us? What carbohydrate do they have on their red blood cells? They have B. And we would classify this person as having B-type blood. What if I saw a reaction in both of those wells? What kind of blood would they have then? AB. What if I saw a reaction in neither of those two wells? O. Okay? Now, so does everybody understand? If you see agglutination, you write that letter down. If you don't see agglutination, you don't write the letter down. And if you get through both of these, A and B, and you haven't written the letter down, you write O. If, however, you write down A, and then you also see a reaction in B, you also write down B. So this type of person here, I would write that person down as AB. Finally, you're asking the question, do they have RH or not? And you're looking for the same thing. If you see a reaction, they do have RH. But we don't write ABRH, we write AB+. If, however, you don't see a reaction, like right here, you write down minus. So if this person instead did have that particular reaction, where we see the agglutination, we write AB positive. Okay. Let's do one more real quick, just so everybody's on the same page, and then we're done. Okay? So, final one here, we'll just do one more example of blood typing. I'm not going to give you all the hints this time. I'm going to have you guys tell me what kind of blood type this person is. Alright, so here we go. What kind of blood type do they have? A negative. Now, the one final thing I want to leave you with is sometimes agglutination doesn't look like particulates. So agglutination either means particles, or it can also sometimes mean that you see an orangish color. You need to see what I mean. Look on the website, on those pictures I have, and you can actually see the color change that much. Alright, so that's it, guys. Good luck studying and have a good Thanksgiving break.